Hello, my name is Konvalinka. I'm a transplant nephrologist and clinician scientist at the University Health Network in Toronto. I care for patients with a kidney transplant, and my research program is entirely motivated by unmet clinical needs. One clinical need that my lab aims to address is how to make the kidney grafts live longer so that every patient would need only one kidney for life. There are three main causes of premature kidney graft loss, and they include antibody-mediated rejection, scarring or fibrosis, and ischemia-reperfusion injury, which is the type of injury that kidney sustains at the time of transplant. And although my lab studies all three of these clinical problems, we're mainly focused on the study of antibody-mediated rejection. I have a PhD in basic science, and I address clinical problems by using molecular profiling, for example, proteomics, which is the study of all different proteins and how they interact in the tissue of kidney, for example, from patients with kidney transplant and antibody-mediated rejection, or in biological fluid like urine or blood. I also use systems biology, which is the study of how clinical characteristics of patients can be integrated with these molecular profiles in order to gain a better understanding of a complex disease such as antibody-mediated rejection. We recently published a manuscript that describes a proteome of biopsies and different compartments within these clinically indicated biopsies from patients with antibody-mediated rejection or other forms of graft injury. And we're the first to describe early changes in the matrix of kidney with antibody-mediated rejection that had not been recognized before. We also identified two proteins that may play an important role in the disruption or remodeling of the matrix in antibody-mediated rejection, and we're studying their biology further. In addition, we're studying how kidney cells interact with immune cells by using sequencing of single cells in patients who have antibody-mediated rejection. We're also studying properties of circulating antibodies and how these properties may incite injury. And finally, we're focused on developing better models to study antibody-mediated rejection in a patient-specific context. To do this, we're working with two biomedical engineers in order to develop a kidney on a chip. Ultimately, the goal of my research program is to improve outcomes for patients through innovative and precision treatments that aim to prolong kidney allograft survival.